So first, are there any introductions? No? Oh, I'd like to make one. Casimiro. Stand up. Stand up. It's Casimiro, who's a graduate student at the University of Sao Paulo, visiting us for a couple of months for work on a collaborative project in the Atlantic Forest. It's a student, Miguel Rodriguez, at USP. Thank you. Any others? Anyone else I can embarrass horribly?
Probably many of us.
Yeah, well, yeah. It's not quite the challenge I was going to rise to. Close. Okay, some seminar announcements. IB this Thursday is Rasmus Grunfeld Winter. Probably mangling that. University of California, Santa Cruz. The genetic reification of race, a story of 3 mathematical methods. Wildlife on Friday is Patrick Kelly, PhD student at UC Davis, on linking altered predator communities to changes in physiology, behavior and demography in neotropical rainforest birds. ESPM on Monday, happily no longer conflicting with research, is Neil Sasui from ESPM, talking about integrating genomics and chemical ecology to illuminate insect behavior. Good one. Now I have an announcement about MVZ lunch this coming week. I've just sent a note around which I hope will get to most of you. And that is, I'm aware there's action plan by students from 12 to 1 on the 9th. Is that? I'm seeing things around campus about it. Yeah. So it's in, and I don't want to conflict with that. I think we should support the students to scream from the rooftops about tuition fee hikes and the 99% thing and all that. So I propose we move MVZ lunch next week, which is Jeff Good from the University of Montana, a great talk. I want to move it to 11 to 12 instead of 12 to 1. But again, there'll be another reminder go around about that next week. Any spare chairs with you? Doris?
One here, 2, 3.
Any other announcements? Oh, there's one here too. All right. Well, of course, it's a great pleasure to introduce Mike Ryan. Those of you who've been around for at least a few years will remember Mike as the last recipient of the MVZ's Grinnell Medal, for which he gave a public lecture. I don't remember if we pinned you down for another talk. You only gave one seminar last time. Anyway, as you're aware, Mike is here as a candidate for the director of MVZ, professor in IB research. Jim gave a very thorough introduction about Mike yesterday, and I don't have time to read out all his honours and let alone his list of publications, but they're awesome. Mike's a true integrated biologist. He's a postdoc in the MVZ as a miller back in the Pleistocene. Well, I'm from the Pliocene, so I can say. And today he's going to tell us about to be or not to be different species, the fickle nature of sexual selection and speciation in some frogs.
Thank you,
Welcome.
sir. Do the lights stay on, or do they?
No, they can't.
Okay, good. You can get them down a little bit.
Can someone push random buttons down on the wall?
Yeah.
Okay, so
I'll get the front ones off. There you go. Good job.
feels like Halloween.
That's good.
So on Monday, I was talking about sexual selection and communication, and one of the things that I was emphasizing is how we need to have an understanding of mechanisms internal to the animal to really understand how it responds to stimulus variation, in that case, courtship signals. And what I want to do now is I want to, so a lot of that talk was about what was happening inside the animal. And what I want to do today is talk more about what's happening outside the animal in the context of sexual selection and speciation. Okay, so we all know that when Darwin wrote on the origin of species, he really didn't attack the problem of speciation very clearly. But this was mostly because of Meyer and Dobzhansky. The idea of speciation became very critical during the modern synthesis of evolutionary biology in the early and mid 1900s. And I think, no matter what anybody's species concept is, whether you agree with Meyer's species concept or not, there's no question of the importance of understanding how speciation works in the understanding of the generation of biological diversity. Maybe it's been overemphasized too much, but regardless, it's been emphasized for quite a long time. Now, lots of studies, excellent studies of speciation have concentrated, I missed a slide. Just disappeared. Lots of studies of speciation have concentrated on ecological aspects of speciation. And in the basic allopatric model, we all know populations become separated. Drift can be important, but the emphasis has often been on ecological adaptation to the local environment, and then as a consequence of this, in Meyer's model, when the populations come back together, there's been drift of the species mate recognition mechanisms. And in Dobzhansky's model, there actually has to be reinforcement. There has to be further evolution of differences in species recognition when animals come back in contact, in Dobzhansky's idea, in his model, for speciation to be complete. But there's another way to view speciation, another mechanism is not the correct word, another narrative for how speciation can take place. And this was an idea that was first suggested by Fisher in his natural selection book in 1930. And Mary Jane West Eberhardt picked up on this with the verbal model in the late 70s. Then Russ Landy presented an analytical model in PNAS in 1981. And the point here is that when you have sexual selection acting within a population, it can cause the divergence of mate recognition signals. So in many cases, the courtship signals that are used to attract females and to compete with males within the population are also the same signals that animals use, that individuals use to choose males of the correct species. So what Fisher and West Eberhardt and Landy suggested is that you can have, because of the random nature of the types of traits that might be favored by sexual selection, you can have divergence of these species recognition characters amongst populations to the extent that if these populations come into contact, they don't recognize one another. And there's some derivative theories of this by a person who I disagree with, so I couldn't believe his name. Isn't that the guy from South Africa?
Patterson.
Patterson, yes. And some of Patterson's ideas are derivative of this, and some of his ideas make sense, and some of his ideas are almost certainly incorrect. Shortly before he passed away, Ham Carson wrote this very interesting paper in PNAS where he suggested that in most cases, sexual selection probably had to be at the basis of the speciation process. I'm not sure that I would agree with that, but that's what Carson suggested. So we went from having very little interest of the role in sexual selection driving speciation to at least a lot of theoretical work suggesting that this was a real likelihood. So what I want to do is I want to talk about some studies that we have done looking at this interaction between sexual selection and speciation. I'm going to talk about two different studies in which the results are just the opposite of what we had predicted. Now, there's a certain number of criteria that need to be demonstrated to make the argument of the importance of sexual selection and speciation. One is that male signals that are used in sexual selection and in species recognition have to differ amongst taxa. The females, or if we're talking about polyandrous systems, when there's male choice of females, this direction of the sex is unimportant here, that the receivers must be attentive, must be able to perceive the signal variation and must influence their mate choice. What's critical here, and it's not always easy to demonstrate, is that this divergence in mate recognition signals between allopatric populations is not due to genetic drift, but that it's actually due to sexual selection. And that once you have the divergence of these signals, this should result in reduced gene flow between the populations because they're no longer recognizing each other as appropriate mates. This is the slide that I thought that I lost. And the point of this slide is just to remind myself that most studies of speciation don't look at this aspect of sexual selection and very much rely on the importance of ecological adaptation, which then result in ecological speciation. And I'm not at all suggesting that that's incorrect or unimportant or very, very common. This has been well demonstrated by many, many researchers, perhaps best recently by Rolf Schluter. So there's no question of the importance and the commonality of ecological speciation. But I think that's probably not the only way that speciation Jesus! This is why I mentioned that gun thing. Holy God.
Sit next to them.
I'm really sorry. I turn this down before I start. Okay. Okay, so these are the tumor frogs that we've been studying, and these are the species that I talked about on Monday. And I talked a little bit about the natural history of these species. They breed in these temporary pools throughout Middle America. They're quite common. They breed in the rainy season, and where we've done most of the studies in Panama. I'm going to talk about other places we've worked on them. And in Panama, the rainy season extends from roughly April or May to the end of November. Now, the district Physalinus pustulosus, and sometimes this genus is referred to as Angistomops. Pustulosus is the only member of this genus that's in Middle America. We're not quite sure how many species there are in the genus. We've collected and worked with somewhere between 35 and 45 species. The largest number of species that we've worked with is in Brazil. But what I'm going to concentrate on here is the Physalinus pustulosus species group. So all of the other Physalinus are distributed throughout South America, the biggest concentration of them throughout the Amazon, although there's lots of Physalinus in the dry temperate forest in southern Brazil, in the Atlantic rainforest on the coast, and then where I haven't worked with them in the temperate beach forest in Argentina, they seem to be fairly abundant. The tundra frog, the northern part of its distribution is in Veracruz, 100 kilometers north of Veracruz. There's a little nuclear power plant, and there's a population of tundra frogs right there. We haven't found any between that population and Veracruz itself, but we assume that they're there. They're found throughout much of the lowlands of Middle America. They're up in here in the Magdalena Valley in Colombia. They're distributed broadly throughout the Llanos of Venezuela. This is the only place where there's sympatric with another Physalinus, Physalinus nsfi, a congener but not a member of the pustulosus species group. They just come up onto the Guiana Shield, and then they're in Trinidad. So a number of years ago, my colleague from Smithsonian, late Stan Rand, Stan and I decided that we wanted to have a better idea of patterns of geographic variation in the taxa. So we did a transect where we left from Austin, and at all of these populations, we collected specimens, we collected mating calls, and we collected tissues. We thought we were collecting the tissues for allozyme analysis, but we've used the tissues for a number of things since then. So again, we have populations fairly evenly spaced throughout Middle America. We've also done more fine-scale transects in central Panama, where we work. Most of our behavioral studies are done in the area of the Panama Canal. And then we have a site from Colombia, a few sites from the Llanos, and a site from up here in Trinidad. And what we found from this, looking at the geographic variation, the phylogenetic analyses, which was first based on allozyme analyses, and this was done with Lee White, who was then at the Smithsonian Tropical Research Institute. We then used sequence of mitochondrial DNA of 16S, and Lee did the lab analysis, but Andrew Crawford, who's hidden in the et al. Here, was very, very important in helping us understand the historical and phylogeographic patterns here. So what we first found just from the analysis is that the tundra frogs break into two separate groups, two separate genetic groups, and that's indicated here. So the tuber frogs, pustulosis, its closest relative is Physalinus petersi. And we meaning Canatella, myself, and Chris Funk, who's involved in various parts of this research, have named split petersi into two species. One is petersi and one is freibergi. For this talk, I'm just going to refer to them as petersi. But these species, they're on the eastern side of the Andes. Petersi, which we'll talk about a lot in this talk, they're distributed in the eastern side of the Andes, and they go all the way to Belém, Brazil. And they're distributed throughout Amazonia with some really interesting gaps, like around Manaus, for some reason. There's no petersi there, and we don't understand why, and the herpetologists in Manaus don't understand why. So this split took place about 10 million, somewhere around 10 million years ago. The other species in the species group, Collardorum pustulatus, and these two then undescribed species, those are all on the western side of the Andes. And there's more species here now. There's 6 species on the western side of the Andes. And one of our colleagues, Santiago Ron, has been doing a lot of work with those frogs. But what's interesting is here, this split, these are all the populations of the tuber frogs that we studied. And the allozyme data show that they fall into 2 separate, we'll call them genetic groups or clades. The mitochondrial DNA sequences supported the same interpretation. Now what's interesting here from a biogeographic point of view is that, remember, pustulosis is a South American genus. These guys are in Panama. This split seems to have taken place somewhere, on the average of 12 million years ago. Yes, around 12 million years ago. What this suggests is that pustulosis probably invaded Middle America prior to the formation of the Panamanian land bridge, if the Panamanian land bridge formed about 3 million years ago. There's some recent geological data that my understanding is, I don't know if it's published yet, but my understanding is very controversial, suggesting that the land bridge actually might have been in place 20 million years ago. But if this interpretation is correct, that suggested that the tuber frogs invaded Middle America prior to the land bridge, and then there's probably another invasion that continued into Middle America over the land bridge. But regardless of the mechanism of dispersal, what we do see here is 2 very large and well-defined genetic groups based on allozyme analysis and mitochondrial DNA sequence. So I had a postdoc who's now at the University of Hamburg, Heike Pruhl, and she studied these genetic groups in greater detail. And what we see is there's an area in here where we don't have any samples of pustulosis. Heike returned and did much more detailed geographic sampling. And there's a 200-kilometer stretch in here where you don't find tuber frogs. And when Stan Rand and I first collected this transect, we were surprised not to see the frogs, but it's nature, this happens. Then he and I had separately returned other times and we couldn't find the frogs. I returned with Heike and there's no tuber frogs there. And Jay Savage, who, amongst other things, a few years ago wrote The Herpetology of Costa Rica, Savage told us that he had known for a long time that there's not tuber frogs in this area, and he doesn't understand why either. But he's also fairly convinced that it's not that we're missing them, they're not there. So Heike did extensive sampling. She used a number of microsatellites that we've developed for the tuber frog. And this shows the results of a structure analysis. And what this also shows, with the microsatellite variation, it seems that the tuber frogs are falling strongly into 2 genetic clades. There's some evidence of some population genetic structure into a 3rd clade, which that phylogeny also suggests as well. The isolation's not as strong, but we're going to concentrate on this more conservative interpretation that there's at least 2 genetic groups here. So this is based on microsatellite analysis. Look at mitochondrial haplotypes, and this is work by Heike Pruhl and Santiago Ron using the same tissues that we had collected years ago. Again, you get the same result, that the haplotype structure suggests that these are really 2 genetically isolated groups. So that's for this. That's the point that we want to make with this phylogeographic and population genetic analysis, that we have what we're calling the species of frog with a 200-kilometer gap in its distribution that appears to perhaps have been separated from one another for perhaps 6 million years. So there's lots of temporal separation within the species, and there's lots of geographic separation between these two groups. So as this, and also there seems to be some ecological separation. When we look at the northern clade of the northern group of the tundra frogs, you tend to find them in drier, more temperate forests in Costa Rica. So in Costa Rica and Nicaragua, Salvador, and Mexico. For people who have worked in Costa Rica, it's very common to find tundra frogs breeding in that very dry forest in Guanacaste. When you see these types of dry forests in Panama, for instance in the province of Chiriquí, you don't find tundra frogs in generally the same kind of arid environment where you do find them in Costa Rica. This is an analysis that Santiago Ron did. If you project the habitat preferences based on moisture back to the last glacial maximum, it suggests that there probably was not a lot of connectivity between these two groups. And Santiago's interpretation is because they're adapted to different environmental regimes. And I'm not sure, and we certainly haven't demonstrated adaptation here. I mean, that kind of adaptation is something that's happening at the physiological level that we could test. I'm not sure that this suggests a hypothesis. I don't think it demonstrates results of selection. So we have these two groups that for all intents and purposes should really be different species, regardless of what your species definition is. So we're interested in sexual selection, so we thought we're getting excited, Stan Rand and I, wow, this is going to be a really nice system in which we can start to study how sexual selection drives speciation. So again, the idea would be that these populations have been separated for quite a while. There's been plenty of time, there's been plenty of lack of gene flow, we think, between the genetic groups, that there should be divergence between the mating signals, and what we wanted to do is quantify that divergence between the mating signals, then ask what degree have we seen the evolution of behavioral reproductive isolation.
So Mark, can I ask a quick question?
Yeah.
So where are we on that original phylogeny you had, the allozymes and the trees? Are we the sort of the basal split or at the...
So we're talking about this split here.
Okay.
Okay. And in that structure model where there is some support for there being three groups, that would be looking at incorporating those two groups. Okay, so we measured the advertisement call for all these populations. There's a set number of acoustic parameters that we measure. And as I pointed out on Monday, we can measure signals, we can measure any kind of a stimuli very easily, very quantitatively nowadays, but that doesn't mean that it's at all perceived by the animal. But what we do know here is when we measure these, we can measure these particular signals, and then we can synthesize a signal that's an average signal for the population based on these parameters. And when we do that, if we give the female a choice between the synthetic call and a natural call, in one case they prefer the synthetic call, in another case they prefer the natural call, and in two cases their preference is 50-50. So the only point is that we think we're capturing salient features of the calls that we measure here, because we can throw all of the other information out, we can make a call, and the females can't discriminate it from a normal call. Okay, do the calls vary? And I could show a graph of one parameter by one parameter, or this is showing these are the calls distributed across the first three principal component axes. And what we see is, yes, the calls do vary. The dark dots here, the dark circles, are from northern populations, and the gray circles are from the southern genetic group. So there's very strong, statistically significant differences between the calls. When you listen to the calls, you wouldn't think they're different species. But if you do this analysis one parameter by one parameter, almost all the parameters are also statistically significantly different. Now, Peter Grant once, in discussing studies of ecological character displacement, pointed out how it's very important not to confuse displacement with clinal variation. And a lot of this variation between the two groups is clinal, but a substantial portion of that variation is not. A statistically significant portion is not clinal. So clinal variation doesn't explain all of the call differences. It does explain a lot of them, but there's significant residual variation that's not explained by clinal variation. Okay, so then the experiments that we wanted to do are quite simple. We went to our focal population, which is this population that we work with in Panama. We synthesized the average mating call for all of the 30 populations that we studied. Then we did 30 phonotaxis experiments. We tested 20 females in each phonotaxis experiment, and we simply determined if the females discriminate between the local call, the average synthetic call of their population, versus the foreign call. And the foreign call could be within their genetic group, or it could be in the other genetic group. We do this for all of the populations. And I explained how we do these phonotaxis experiments on Monday. They're fairly simple. We collect amplex animals in the field, bring them in an acoustic chamber. There's two speakers. They're given two alternatives, so they would hear, ear, ear, ear. We don't use the chucks in this analysis. We're only using the whines. And then these are the results, and the results were very disappointing. So this is a 50-50 preference. This is the null hypothesis. Given the sample size of 20, if more females than 0.7, I think it's 0.77, went to the local call, this would be a local preference. If fewer than 0.23, fewer than 23% of the females went to the local call, that would be a significant preference for the foreign call. And if the strength of the female preference is between these two parameters, these two lines, then that's statistically not a significant preference. This dotted line here shows the populations from the northern group versus the populations from the southern group. So what we see here is that in only a few cases are there strong significant preferences for the local call. Only in a third of the experiments did females prefer the local call to the foreign call. There's significant preferences amongst populations, meaning amongst the calls that the females respond to, but there's no significant preferences when the data are partitioned by genetic group. So there's no hint here whatsoever that despite 6 million years of isolation and 200 kilometers of separation, that there's been any divergence of these mate recognitions. So Charles, you have a question?
Are there size differences? Could they just be straight volume selection as opposed to structure of the call or something? Did you look at that?
Well, we did. There is variation in body size, and the variation in body size is cardinal. When you analyze the calls for geographic variation, if you use body size as a covariate, the calls are still significantly different from one another. So that doesn't explain the difference in the calls. If body size, go ahead.
But are they selecting for, say, different volume rather than actually the...
You mean volume of the call?
Yeah, so when you did your lab experiments, did you actually have volume?
No, we standardized the call amplitude in the experiment.
So they could be picking up on body size.
Well, no, in the synthetic calls, we play them all back. We broadcast them in the testing chamber at the same amplitude.
Right, but in the wild, they could be...
In the wild, yeah, but they could, and they do in the Gamboa population by differential preference for dominant frequencies of the signal. Even if larger males call louder, it's not a reliable cue of body size because amplitude attenuates with distance. So, unless you have a mechanism to what's called ranging, which we actually think that these guys do. But yeah. So it is possible that there's a preference for body size, but it wouldn't explain this lack of pattern. So we thought this would be this wonderful case where we could, jump feet first into these, very detailed empirical studies of sexual selection and speciation, and there's just nothing there. Except we thought maybe what we need to do is concentrate in the area of contact. I mean, the two genetic groups don't come into contact. They're separated by 200 kilometers, but I'm just going to call that the contact area, the contact zone, for lack of a better word. So again, Heike worked extensively in this area, and again, she sampled more populations and did this microsatellite analysis, which again gives us the same genetic structuring. But then what she also did is she tested, this is a little bit differently, she used synthetic culls, and she tested the preferences from females from many different populations. So what she would do is she would test a female, and she would give it a choice between, say from here, between its local call and the call from Liberia, its local call in Golfito, its local call in Garichi, its local call in Santiago, its local call in Gamboa. And if a female had to respond in all of these different phonotaxis experiments for us to include the data, because we analyze the data with a repeated measures logistic regression analysis, which gives us a lot more power. And so this is what we found. This is the percentage of females who preferred the local call versus the foreign call. And what we see here is that in this case we have about two-thirds of the cases their preferences for local calls. But there's still no suggestion of any preference based on genetic group. So this is the only one in the northern group, but all, with the exception of Gamboa, these populations are all in the southern group, but these are the closest populations to the northern group. So again, even when we zero in on the area where we would expect selection to be acting strongest to cause divergence of the mate recognition signals, there's no hint. There's no hint that that's happening. Then another question we asked, getting a little bit frustrated, where, biogeographically, these things should be diverging. So this made us step back and just ask a very general question. What are our expectations? What's the null expectations? To what degree are mating calls between populations diverging as a function of time and as a function of genetic distance? So Owen and Kaur published this very important paper in 1989 in Drosophila, and what they did is they examined the degree of prezygotic and postzygotic isolation as a function of genetic distance. And what they showed is that you do get this pattern. The measure of genetic distance here is Ney's genetic distance. As Ney's genetic distance increases, then so does their measure of behavioral reproductive isolation. And they measure this in Drosophila quite differently than people do in other systems. But they argue that that this is a trend that we should see in other animals. So we did a lot of other experiments, and we asked, on the very broad scale, can we make any statement about the degree to which signals and preferences diverge as a function of time, as a function of genetic divergence? So the first question we asked, and this is, we wanted to know, within a population, is females' mate choice biased by the degree of relatedness, measured with microsatellites, by the degree of relatedness to other individuals? So is there inbreeding avoidance or is there outbreeding avoidance? And what we found with very large sample sizes, much larger sample sizes than previous studies have looked at inbreeding avoidance in other animals, that in these frogs there's no hint whatsoever of inbreeding avoidance. I just showed you the data from amongst populations, and there's also no hint that female recognition, female preferences for mating calls scales in any predictable manner to the amount of genetic divergence. And now we included with that preferences for calls of the conspecific versus the different species, and we see here that within here there's no statistically significant relationship. Now if one were to analyze all of these data together, there would be a trend, but this trend is because there's no trend here. The preferences are random within a population and among populations, and then when you go to species, the preference plateaus. That in most cases there's unanimous preference for the conspecific versus the heterospecific signals. So, looking at sexual selection, looking at spatial and temporal divergence at different timescales between preferences and calls, we're really frustrated here, but this is what we found. And this isn't what we learned from the textbooks about how mate recognition systems are supposed to diverge. But most studies of divergence of mate recognition systems are looking at taxa that have been differentiated into species for quite some time. And we thought it would be important to look at incipient speciation taking place, because that's what I think we'll probably see. So that's the story with the tuber frogs. There's just no evidence for this. This is the sister taxon to tuber frogs. This is Physalimnus or Angistomops petersi. Again, it's distributed quite broadly from Ecuador down the Andes into Bolivia, across through the Amazon, and then we've worked with populations in Brazil and Belém. So it's widely distributed. We actually don't have a very good understanding of its geographical distribution. Most of it, as I'll show you, all of the work has been done in Amazonian Ecuador and Amazonian Peru. So the populations, the studies that I'm going to talk about, again, take place in Amazonian Ecuador, and we're going to concentrate mostly on two populations, La Selva and Yasuni. And these populations are on either side of the Rio Napo. And for people who have been on the Napo is not a very imposing geographical barrier, depending on where you are. There's when I first started this research, I worked down here in Hatun Sacha, where the Physalimnus petersi have only simple calls, and there's times of the year where you can just walk right across the Napo. If we look at Yasuni and La Selva, they're in very similar habitats. So the first work that I'm going to talk about here, as I said, I worked in Hatun Sacha for a while, and the Physalimnus petersi make only simple calls. And an undergraduate came into my office and she said, I'm doing this exchange program, I'm going to Ecuador, I like animals. Someone said I should talk to you to see if there's a little project I should do. I said, yeah, there is. Here's a tape recorder. I said, there's this place called Hatun Sacha, and I worked there for a while, and these Physalimnus petersi do not make complex calls. But one of my former students, Rex Cocroft, and this herpetologist Roy McDermott, they had worked in Tambalpato in Peru for a while, and for 3 years they recorded tuber frogs, and there were no complex calls. And then I said to Rex, bring down some tuber frogs calls with chucks to play to the petersi in Tambalpato, and then they respond with chucks. So I armed Kathy not only with a tape recorder, but with records to play, calls to play back. So she went to Hatun Sacha, where I had worked maybe 5 or 6, 7 years before, and still the Physalimnus petersi there don't make chucks, and they've been surveyed many times, and they don't make complex calls. Then she went to Yasuni, and then what she found in Yasuni is that they do make complex calls. So I'm going to play you. Remember, La Selva and Yasuni are just opposite one another on either sides of the Rio Napo. They're separated by only 20 kilometers. And this is the advertisement call of the males from La Selva. They only make a simple call, just a simple whine. This is the call from Yasuni, and the males from Yasuni, like the tuber frogs, they can make a simple call, or they can add this other syllable to their call. And in the literature, we refer to this as a squawk. But I'll show you later, I'll explain later why we think it's homologous to the chuck. But it sounds differently. So I'll play these calls. You ready? I'm trying to sound really loud. Only because the calls are really soft. But you want to hold on to this? Okay. So this is the simple call from La Selva. And this is Yasuni. It sounds quite different, but it's a simple call. And now this is what they sound like when they add the squawk or the chuck. Isn't that wild? I'll play it again. I'll play it one more time, only because it's so beautiful. And the males add these syllables on very similar to the way the tuber frogs do. When males are calling by themselves, they make only this simple call, and then when you challenge the males with the conspecific call, they add the squawk. One way that they're different from the tuber frog is they can only add one. And this is a much longer sound than the tuber frogs. And probably, I'm sure you'd agree, Robert, they're just expending all of their air making this squawk, and that's why they can't make more chucks. Okay, it ends up that this pattern of having species next to, of having geographically close populations that have diverged that have simple calls versus complex calls in different populations has evolved several times. It's evolved here between Yasuni and La Selva, and then it's evolved in these other populations here. And we now know of at least 3 or 4 other cases where there's 2 populations near one another. One can make complex calls, where the other population makes only simple calls. And this coalescent analysis here was done by Chris Funk, who did all the population genetics research that goes along with this study. So, first question that we asked is, in La Selva, where they have complex calls, do the females prefer the complex, in Yasuni, where they have complex calls, do the females prefer the complex call to the simple call, as do the Tungura frog females? And the answer is yes. Kathy tested 15 males to 15 females. 13 of the females preferred the complex call, and only 2 of the females preferred the simple call. Now in La Selva, they don't produce complex calls, but what we're able to do is to add the squawk from Yasuni onto the whine of La Selva, and what we see is that there's not a statistically significant preference for the complex call versus the simple call, although the power to accept the null hypothesis is pretty low. So there's a majority of the females prefer the complex call, but it's not statistically significant. But this is asking a question more about sexual selection within populations, and what we want to know about is behavioral reproductive isolation. So the whine is the species recognition component. So what we want to know is, how do the females respond when they have a choice between their own whine and the whine of the nearby population? And what we see is pretty amazing. Females from Yasuni have a 100% unanimous preference for the local call versus the call of the population only 20 kilometers away. And just to remind you, this is based only on simple calls, not including the squawk. When we test the females from La Selva, they also have a very, very strong preference for local calls versus foreign calls. 17 females prefer the local call, and only 2 females prefer the call of Yasuni. So what we see here now is 2 populations quite close to one another. And again, I make the argument, and I can't prove, but I'm making the argument that the Napo is not a formidable geographic barrier. And the sequence divergence between these 2 populations is only 1/2 the average sequence divergence between the northern and southern Tungura frog groups. So there's been a lot less genetic divergence, as one would expect, between this pair of populations than between the 2 big groups of the Tungura frogs. And yet what we see is that at the level of behavioral mate choice, these animals are acting as if they're different species. They have very strong and total preferences for a local call. And this is exactly not what we would expect, right? We would expect sexual selection would have been driving the divergence of these groups that are genetically, temporally, and spatially more isolated from one another. In these populations, where you can almost throw a rock from one population to the other, there's been this divergence. We're certainly not sure that there's been no ecological selection. There's nothing obvious to us. Chris Funk has measured the presence of these frog-eating bats at the 2 populations. He's able to put out speakers, then just put remote cameras, and you see the trachops flying in, catching frogs. And there doesn't seem to be a difference in the relative abundance of trachops in these places. Although he hasn't netted the bats. He's just done this. Okay, so what's going on here? These animals are pretty similar in many, many aspects. Their calls are kind of similar. You know, some populations can make complex calls, some can't. Ecologically, these populations are similar. They're not this different than the Tungura frogs. When you're working with these animals in Ecuador, they're a little less clumped in space than Tungura frogs. There are some differences. Quite similar animals. What's going on? Well, they're only similar if you look at them from the outside. If you go inside these frogs, then you uncover, it's like opening an oyster and seeing a jewel. Looking at the larynx of these frogs is just an amazing experience. So here's their larynx. So frogs have a voice box very similar to ours. One big difference is we have a descended larynx, which most mammals don't, but we do. The frog's larynx sits right up on the top of the trachea, and if you move the frog's tongue, you can look right into its larynx. You can kind of open it up and see the vocal folds. As with the mammalian sound production system, the lungs are the power supply for making the call. The frogs don't really have a bronchus, so the lungs attach right to this bronchial process. The air enters here from one lung and then into the other half here. This is called the arytenoid cartilage. The air comes in here and it vibrates the vocal folds. As the vocal folds vibrate, the arytenoid cartilages open up, kind of like ours. The opening can be passive due to a pressure buildup that then just causes them to come apart. But there's huge muscles, especially in the tundra frog. The muscles are very large that surrounds the arytenoids. And if you contract those muscles, if you stimulate those muscles, you can see the vocal folds, the arytenoid cartilages pop open. So the sound is this. It's the vibration of the vocal cords that then cause the sound pressure disturbance that we hear as sound. And then there's some other difference between frogs and mammals. We open our mouths when we talk, and frogs keep their mouths closed when they talk. And that's why they have a vocal sac, so they can move all that air, use the same air back and forth, because they don't have a diaphragm. So they have positive pressure breathing, so they have to do this buccal pumping to fill their lungs. So they just, the vocal sac appears to have evolved to allow the shuttling of air back and forth. But this is the weird thing about the vocal cords. It's this thing here. This is a part of the vocal folds, the vocal cords, but we call it it's a fibrous mass. So it hangs from the vocal folds. It's very large. And if you look from the perspective of the lungs here, you see it pretty much occludes airflow. It's a pretty important structure. Now, years ago, Rand and Drury and Heyer compared the laryngeal structure of the tundra frog to two other species of Phycelemus that don't produce complex calls. And what they showed is that the fibrous mass is quite reduced in species that don't make complex calls. These fibrous masses are found within the subfamily Leptodactylinae, to which the Phycelemus belong. They're also kind of common in the family Leptodactylidae. But even when you don't have masses, frogs make very deep calls for their body size. A tundra frog is this big. The wavelength of the lowest frequency it makes is this big. If you know acoustics, you know that doesn't work. How do you do that? Well, what they do is they hang these kind of testicle-like things from their vocal folds, or if you look close here, you can see there are these thickenings of the vocal cords that add weight to the mass of the vocal folds, and that's a very common trick in frogs. This is bizarre, to have this big thing hanging off your vocal fold. So the correlational data from Heyer, Drury, and Rand suggested a structure-function relationship. Then Bob Drews of Calicut and I worked with the collection in Kansas for a while, looking at other Phycelemus in the species group, and we found the same pattern. The other Phycelemus, especially on the western side of the Andes, have a very, very tiny fibrous mass. So it appears that the fibrous mass is implicated in production of the chuck. Then I had this wonderful Brazilian grad student who stayed around for postdoc, Marcos Gritty Pop. And what we wanted to do is we wanted to show experimentally that the fibrous mass was involved in chuck production. So if you look here at the real larynx, you say, well, that's pretty easy. Just take it and cut it off, and then have the frogs call. Except the larynx isn't this big. The frog's only this big. So what Marcos was able to do, we recorded calling tundra frogs in the field. We recorded a male till he produced a chuck. We anesthetized the frog, and then because the larynx opens up into the frog's mouth, Marcos was able to go in to the larynx, pull out one vocal fold, cut off the fibrous mass, the other vocal fold, cut off the fibrous mass. Then he did the appropriate sham operations. You just take the scissors and rub it against the fibrous mass. Then eventually the frogs would recover from anesthesia, and then eventually they would call. And then what you found, what you found is that the frogs would, in a sense, try to produce a chuck, but instead of sounding like this, they would sound like this. So for the frogs to produce a chuck, Robert Dudley and Stan Rand showed this, there's a further contraction of the lungs that sends this burst of air through the larynx. So the whine decreases in amplitude. So they're contracting their lungs again, putting this air through. So that's voluntary behavior. But what happens is all they make are upper harmonics of the whine. These chucks have a fundamental frequency that's half the fundamental frequency of the whine. And this ends up being interesting from what we know about mammalian sound production. What's happening here is that we think the fibrous mass is moving inside this bronchial process, and it's probably hitting the top of it as it vibrates. And I'll talk about some work we're doing with some engineers, and the engineer thinks what's happening is this is called clapping oscillations. So you introduce a lot of nonlinearities into the vibration of the vocal folds. In mammals, and especially monkeys, this is quite common. So you can go from a harmonic series to what these are, a subharmonic series, and then if you drive it further, and again, it's well demonstrated in primates, then you get chaotic behavior of the vocal folds. So it appears that the frogs are stopping just short of chaos. But more importantly, what it shows experimentally is, yeah, that fibrous mass is critical for producing the chuck. Let's go back to the Peter's eye. If we look at the larynx of the two populations of Peter's eye, the La Selva does not make chucks. This is from the western side of the Andes, Physalis coloradorum. This is from Ecuador, Condo. They don't make chucks. Peter's eye from Yasuni makes chucks. Peter's eye from Tambopato in Peru makes chucks. And Chungur frogs make chucks. If you compare the area of the larynx, the area of the fibrous mass, this is, I think, yeah, this is just a different measure of the area of the fibrous mass and the size of these bronchial processes into which the fibrous mass produced. The point is, what you see is that the Peter's eye that make squawks or make chucks have a larynx that's quite similar to the pustulosis larynx, quite different from their neighbors 20 kilometers away. And those frogs from La Selva and other populations where they don't make chuck, they have a larynx that's less like the larynx of their conspecifics and more like the larynx of frogs that reside on the western side of the Andes. So the correlational evidence here shows, again, that this chuck is tied to variation in vocal morphology, and we're seeing vocal morphology at this very fine geographic scale in Peter's eye that seems to be implicated in divergence of in reproductive isolation. But how do we go from this thing about the chuck to the females having preferences for one whine versus the other whine? What happens is, when the males evolved the morphology that allowed them to produce the chuck, not only could they make a complex call, but they had a bigger larynx. So the frequency of their whine was lower. And if you remember that the whines of those two populations sounded different to one another, it's because one was lower frequency and one was higher frequency. And we know that the frog auditory system is very sensitive to differences in frequency. So what our scenario, what our narrative is that in Yasuni, for some reason, males evolved the larynx, the fibrous mass, that allowed them to make complex calls. This was favored by sexual selection because the females preferred the complex calls. So this morphological mutation was maintained by sexual selection. An incidental consequence of this change in the larynx that was favored by sexual selection was a decrease in the frequency of the whine, making it different from the frequency of the whine in the nearby population. Now that narrative could be turned around in the opposite direction. We don't know the polarity of the changes here, but either way, it will go either way. Okay, so what do we want to, and then my current graduate student, Monica Guerra, who worked with Santiago Ran when she was at Universidad Católica, she's in my lab now, she worked with another population in Puyo, and she repeated these same discrimination experiments, and she showed the same thing. There's strong discrimination between Yasuni and La Selva, and there's also strong discrimination between Puyo and La Selva and Puyo and Yasuni. Monica's collected a lot more data since then, as part of her PhD thesis, and what she's finding is incredibly strong preferences for local calls versus foreign calls are the same conspecifics, what we think are the same species, all throughout this area of the Amazon. And she's starting to work in Peru now, and at least the recording she gets from Peru, she thinks that the same thing's going to happen. There's many replicated instances of these complex calls evolving, or of populations losing the ability to make complex calls. And what's happening all along, going from Ecuador down into Peru, is all these populations are all acting like they're their own species. And I don't know of other taxa where we're seeing this very strong degree of population-based preferences. So, well, this is what we want to do now. I just want to briefly tell you about 2 projects that this has spurred. We would like to know, there's a big interest in speciation gene, and many years ago, Chung-Yi Wu said that he had found the speciation gene. It was the genes that control HCH production, cuticle hydrocarbons in Drosophila. Well, he's absolutely right that in Drosophila, those might be important in species recognition. But, I mean, different animals are different. There's certainly no speciation gene, but some people have started using the term magic genes. And what Maria Servideo and other people mean by that is you can have a simple genetic change, simple mutation that then results in the divergence of mate recognition systems, and bam, you kick off the speciation process. This is very different than what Meyer and Dobzhansky envisioned as general differentiation of the genomes of 2 populations. So the larynx in Tungra frogs is dimorphic. So if we look at the size of the larynx during ontogeny, this is about as small as we can operate on the frog and see the larynx after they metamorphose. See, the males and the females have about the same size of the larynx as a function of their body size until the males reach about 16 millimeters. Then bam, huge increase in sexual dimorphism. The male larynx gets very large, and this is the size at which the males start to call. So we're starting a project with, we just started this project with John Wallingford, who's a molecular developmental geneticist at Texas, and Scott Hunnicky-Smith, who runs our next-gen sequencing facility at Texas. So what we want to do is we want to try to uncover what are the patterns of gene expression that are responsible for this dimorphism in the Tungra frogs. And when we're able to do that, we then want to do 2 things. We want to examine patterns of gene expression in the population of the Peters eye that makes simple calls and complex calls. And then parallel to that, Wallingford's fairly sure that once we understand how the patterns of gene expression are important in promoting the dimorphism, and we think the size of the fibrous mass, that he'll be able to regulate the amount of gene expression. So we'll be able to choose males, and John's in the process of making transgenic Tungra frogs now. What we'll be able to do is mutate the larynx to whatever degree we want. So not just look at the larynx, the larynges that we have now that make simple calls or complex calls, but in a sense explore the genotype-phenotype universe for these larynges that would be possible. Then let the males call. Then take those calls and ask to what degree those calls are salient, that they influence the female preferences. So we've just done the first round of NRA-seq on larynges of males that are from right about here. We don't really have any, we have the transcriptome data, we don't have any data analyzed yet. I have some guesses at what some candidate genes might be. Elastin is a gene that's responsible for a lot of the membranes in our vocal folds for the elastin. And we know that point mutations in elastin have very deleterious effects on human speech patterns, and then have cascading effects on the later social behavior of the individual due to the fact that they can't articulate very clearly. And we're all interested in money, so we're hoping that this elastin will work out, because we're trying to convince NIH that this is a good model to look at vocal deformities. And what's very convenient is that Xenopus, which is a better developmental model, don't have vocal folds. So they're a frog they're not really a frog, but Xenopus is a frog without vocal folds, which is a little odd. There's other genes that are probably important. BMP is very important in regulating the development of the size and the shape of the beak and Galapagos finches, And my colleague Wallingford is thinking BMP is a very good candidate. There's a gene, FOXP2, that was uncovered as the human language gene many years ago, overinterpreted quite a bit. But FOXP2 maps on very well to the evolution of echolocation in bats. In bird song, it's expressed when birds are learning songs, so people now think FOXP2 might even have something to do with perception. But the point is, as you all know, with this approach, we're going to find lots and lots and lots of candidate genes, and our hope is that, you know, one of them will make sense, and then we'll be able to examine these populations of Peter's eye. Then the next thing that we're trying to do, which is related, is we're trying to make an analytical engineering model of sound production in the tuner frog for kind of the same reason that we're doing the developmental genetics. Now, in theory, it's pretty easy to sketch out a model of how the vocal system works. There's lungs, there's vocal folds, there's a mass hanging off of the vocal folds. This comes into the buccal cavity, the mouth, and then it goes into the vocal sac, which is elastic, so which has tension that varies as a function of how full it is. And that's about as far as I could get on this model. So I'm working with this engineer, Preston Wilson, who's an acoustical engineer at Texas, and a former grad student of mine, Nicole Keim, who's mathematically quite gifted and a very good programmer. So they're using this approach called bond graph theory. And basically what we're starting to do is to parameterize all the different parts of the vocal production system. We're starting to do a lot of measures of the size of the lungs, estimating the pressure of the air coming out of the lungs, the elasticity of the vocal folds, the tension of what we model as a spring that's holding the fibrous mass to the vocal folds, even though it's not really a spring. And they're starting to make some progress on all the math. So once the math is done, one of the projects that we'd like to do resembles this. Years ago, Stan Rand and I examined the calls of extant species of frogs, and then using these 7 parameters that we use to describe the call, we use various phylogenetic techniques, which are very old now, to estimate those parameters at ancestral nodes. We did that for all 7 call parameters. Then we reconstructed quote-unquote ancestral calls, and then